Welcome to the BS3336 presentation by Group 3. I'm Darren and my colleague Reuben will be giving a video presentation on post-translation modification. This video will focus on carbamylation. Without further ado, let us begin. In this video, we will be covering the following. Firstly, we will present a brief introduction to post-translation modification, or PTM for short. Next, we will cover carbamylation. Here, we will look at its chemical process and its mode of occurrence. We will then examine its involvement in some physiological process. For the remaining segments of the video, we will look into the experimental methods to detect carbamylated proteins. One such example will be covered is HPLC coupled with tandem mass spectrometry. Lastly, we will now perform result analysis and data mining. Now, let me bring you into the world of PTM. From this schematic drawing, we can see the central dogma of biology illustrated here. DNA can undergo replication to produce more copies of itself. Alternatively, it may be transcribed to produce RNAs. Increasingly, transcriptomics is a rising field in biology and many scientific papers have been published. However, in this video we will not focus much on this. Lastly, proteins can be translated from mRNAs. It is known that there are approximately 3 billion base pairs in the human genome. Out of these, only 2% are protein-coding genes and code for about 20,000 proteins. These proteins can undergo PTMs such as phosphorylation, glycosylation, and acetylation, which therefore increases the functional diversity of proteins. No wonder PTM is a very popular field in biology. PTM also play an essential role in many cellular and molecular processes including cell signaling, cell communication, and signal transduction pathways. It is also involved in the folding of proteins and controls the functional states of enzymes, which is to either activate or inactivate enzymes. Lastly, the consequence of post-translational modifications is great, as it can result in the change in protein structure and therefore function. The modification can be enzymatic or non-enzymatic. Non-enzymatic PTMs can be generated by spontaneous chemical reactions. If these modifications result in the loss of function of proteins, they are said to be degenerative protein modifications, as it may cause aging and various degenerative disease. Now, let us look into greater detail one such non-enzymatic modification, carbamylation. Carbamylation is an irreversible non-enzymatic process. It mainly occurs at the amino group of the side chain of lysine residues. Although carbamylation is not enzymatic, it does not carbamylate lysine residues at random. There are preferential sites for carbamylation. Carbamylation is driven by the compound isocyanate. Isocyanate is derived from cyanate through a reversible reaction as shown. When the lysine residue is carbamylated, it becomes a different compound known as homocitrulline. In a protein, which lysine residues are carbamylated is determined by a number of factors. The extent of carbamylation is not directly related to the number of lysine residues within a sequence. Other factors such as accessibility can influence carbamylation as well. At this point, you may be wondering that how is cyanate derived in the body? Urea is a source of cyanate in all individuals and is present in body fluids in equilibrium with ammonium cyanate. Cyanate and urea exist in an equilibrium with a ratio of 1:500,000. Despite the low concentration of cyanate, trace amounts of carbamylation can still be detected in healthy individuals. From this, we understand that patients with renal dysfunction and elevated blood urea nitrogen levels will experience higher levels of carbamylation as urea and cyanate exist in equilibrium. When the kidney is no longer able to perform its intended function of excreting urea begins to accumulate. Therefore, for the equilibrium between urea and cyanate to be maintained and balanced, more urea will have to be converted into cyanate. To understand this better, let us now take a look at the urea cycle. The main purpose of the urea cycle is to remove ammonia from the body, as accumulation of ammonia can be toxic. From the diagram on the right, we see two mechanisms available in humans for the transport of ammonia from the peripheral tissue to the liver for its ultimate conversion to urea. The first mechanism requires an enzyme known as glutamine synthetase to combine ammonia with glutamate to form glutamine. The second transport mechanism, used primarily by muscle, involves transamination of pyruvate to form alanine. This transamination step involves the transfer of the ammonium ion from glutamate to alanine. Alanine is transported by the blood into the liver, where it is converted to pyruvate again by transamination. In the liver, the ammonia is then converted finally to urea and excreted. Therefore, increased production of ammonia will result in an increased product of urea. Therefore, it is noteworthy to understand where ammonia is derived from. Other than from the muscles, ammonia can come from the brain and from the gut. The glutamine derived from the brain, muscle, and other tissues releases ammonia for urea synthesis. From this, we can draw a link or a relationship. Whenever there are high levels of ammonia production in the body, there is higher urea production. Therefore, there is a higher chance of carbamylation occurrence, especially if the patient has some chronic kidney disease and is unable to clear the urea produced. Inflammation is another factor which can stimulate carbamylation. Inflammation triggers carbamylation via a mechanism which depends on myeloperoxidase, or MPO for short. MPO is mainly stored in granules of neutrophils and are active during the respiratory burst to combat against infection. This process generates cyanide using hydrogen peroxide and thiocyanate as substrates. It has been suggested that inflammation has a link with coronary heart disease. When there is a buildup of fatty and cholesterol-rich plaque in a blood vessel, the body's immune system may perceive this plaque as foreign, hence will try to attack it. This triggers the inflammatory response. In response, the plaque may rupture and cause blood clot formation. Formation of such clots can block blood flow especially to important and vital organs, causing cardiovascular disease and stroke. Therefore, patients experiencing chronic inflammation and high levels of oxidative stress may experience higher levels of carbamylation. Since inflammation activity increases, the amount of cyanide present in the blood. Protein carbamylation is thus becoming recognized as an additional contributory link in the pathophysiology of cardiovascular disease, warranting further investigation to develop novel therapeutic strategies for prevention and treatment. Cyanide in air can be derived from various sources such as biomass burning, coal burning, biofuel usage, cooking, tobacco usage, and wildfire. Diet can also influence the amount of urea produced, especially if the diet is rich in proteins. As shown previously, the byproduct of protein catabolism is urea. Smoking can also increase in the concentration of thiocyanate in the body. Next, we shall see how carbamylation is related to some disease experienced by men. Patients with chronic renal insufficiency are known to exhibit repeated infection disorders in the renal cells. Research has shown that this is due to carbamylation of type 1 collagen. Since these patients have renal dysfunction, they experience high concentration of urea. Therefore, cyanide concentration increases, resulting in a carbamylation of collagen. When collagen is carbamylated, the ECM becomes disorganized as shown in the diagram, and the inflammatory response is impaired. This is because collagen is a component of the basal lamina of the extracellular matrix. It stabilizes the integral membrane proteins on the cell membrane to allow cell signaling for activation of neutrophils. This diagram shows that without carbamylation of collagen, neutrophils are activated as cell signaling can occur since the ECM structure is preserved. When carbamylation occurs, neutrophils are not activated. Therefore, carbamylated collagen can be used as a potential biomarker for patients suffering from chronic renal insufficiency. Another example of a disease would include arthritis. Antibodies against citrullinated protein antigens have shown their relevance for the prognosis and diagnosis of rheumatoid arthritis. Citrulline is formed from arginine by a process known as citrullination. Homocitrulline also highly resembles citrulline in terms of structure and is formed through the carbamylation of the lysine residue. Research shows that carbamylation provides the missing link between citrullinated peptides and the development of arthritis. Homocitrulline may be regarded as foreign entities within the body and therefore be targeted by the immune system. This results in the proliferation and chemotaxis of CD4 plus T cells, the production of pro-inflammatory cytokines such as interferon gamma and interleukin 17 and antibody response. This causes autoimmune disease and inflammation, and the local recognition of citrullinated peptides within the joints, ultimately leading to arthritis in an individual. Because of the significance of carbamylation in human disease, it is important to study its effects further so that therapeutic methods can be developed. However, it has been a great challenge to study PTMs since it cannot be determined by genomic sequence. Though genetic experiments such as knockout or knockdowns are often conducted to study the function of genes, it will be difficult to rely on these techniques to understand the functions of PTMs since PTMs are determined by environmental signals. A different approach has to be utilized to study proteins and identify PTMs. In the proteomic approach, we are applying the discovery-driven approach, which studies all the protein in a sample. Since our topic of interest is carbamylation, we will identify proteins which are carbamylated. From there, we can derive a hypothesis and conduct the conventional hypothesis-driven experiments to test and prove our hypothesis. For this video presentation, we will describe how the discovery-driven approach is carried out. There are two ways to do so: the bottom-up approach, also known as shotgun proteomics, and top-down approach. In our experiment, we have conducted the bottom-up approach to identify the carbamylated proteins. The bottom-up approach is an experimental technique that digests proteins into smaller peptides, after which high-performance liquid chromatography, or HPLC for short, will be conducted for fractionation and pooling. The samples will then be run on tandem mass spectrometry to obtain the identity of the peptides through peptide sequencing. In our experiment, we have used the plasma sample extracted from a pig that is suffering from cardiovascular disease. Pigs are good animal models to study the cardiovascular condition which normally occur in humans. The following segments of this video will briefly describe some key steps involved. This will be explained by Reuben.
Trypsin digestion was performed. Trypsin is a protease that cleaves proteins into smaller fragments. The enzyme recognizes the amino acids arginine and lysine and cleaves at the C-terminal. Consider a protein that's the following sequence. This diagram shows that when a protein has an extended conformation, the cleavage sites of lysine and arginine are exposed, which will allow complete cleavage by trypsin. This will give rise to different peptide fragments as shown here. However, this is seldom the case, as the protein may fall into a closed conformation. In this conformation, the protein has folded into a closed loop. As before, two of the amino acids, one lysine and the other arginine, can be cleaved by trypsin. However, one of the lysine residues is no longer accessible for trypsin digestion since the protein is folded. Therefore, we get the peptides as shown here. Instead of 4 peptides, we now get only 3. This is important to note, especially when it comes to the data analysis and peptide sequencing step, which will be discussed later. We should not assume that trypsin digestion is always perfect, and we must factor in the possibility that there could be missed cleavages. After trypsin digestion, the pig plasma was subjected to reverse phase HPLC fractionation to separate the peptides in the pig plasma sample based on hydrophobicity. C18 columns were used. These columns will interact and bind with the hydrophobic peptides. Hydrophilic peptides will pass through easily. In contrast, normal phase HPLC column has no hydrocarbons attached to the beads. These columns will interact with hydrophilic molecules instead. After the fractionation has completed, a total of 29 tubes were obtained in our experiment. Each tube contained different peptides. Tube 0 was used as a blank. Tube 1 will contain peptides which are most hydrophilic, while those in tube 29 will be most hydrophobic. This is the chromatogram that was obtained from the HPLC fractionation. Each peak corresponds to a peptide that was obtained in the fractionation. As the time proceeds forward, the percentage of buffer B added increased, while the percentage of buffer A added decreased. At the 25 minutes mark, 100% of buffer B was added. Since buffer A is more hydrophilic relative to buffer B, peptides eluted out earlier are more hydrophilic as compared to peptides eluted later. After 28 minutes, all the peptides should have been eluted out from the original pig plasma sample. After which, we perform pooling via the concatenation method, where tubes 1, 11 and 21 will be put together, tubes 2, 12 and 22 will be put together, and so on. This was done such that a total of 10 tubes remain. This reduces the total number of tubes that will be analyzed through tandem mass spectrometry, or MSMS for short. The peptides in each of these tubes will then be ionized so that they acquire a charge. This step is important for the MSMS method, as there is a magnetic field generated within the machine which will deflect the charged particles. This ionization can either occur in vacuum or under atmospheric conditions. Soft ionization techniques need to be used to ionize a biological molecule such as peptides. To do so, protonation is carried out and all amine groups in the peptide will be protonated. Let us consider this protein which was shown earlier. After trypsin digestion, we get the peptides shown here. Now, let us use the second peptide as an example. If we conduct protonation, the amino groups will become protonated and each group acquires one positive charge. In this case, the N-terminus group and the side chains of the two lysine residues will acquire the positive charge. Now, the peptides will be inserted into the mass spectrometry machine for analysis. There are three modes in the mass spectrometer. The first mode is the mass scanning mode. Here, the direct current voltage is continually increased to allow peptides of different mass to charge ratios to pass through. The DC voltages specific for each protein or peptide will be known and recorded. The second mode is the single mass transmission mode. Here, a specific DC voltage can be selected to allow one peptide to pass through. The remaining peptides will not be able to pass through and reach the detector in the mass spectrometer. The third mode is the radio frequency mode. It is the mode where all peptides are able to pass through when the DC voltage is set at 0. Now, with these three different settings, the tandem MS approach can be utilized to identify the peptide sequences. Now, our ionized samples are fed into the machine. In the first step, which is to scan the precursor ions, Q0 to Q2 are set at radio frequency mode to allow all peptides to pass through and reach Q3. Q0 simply serves as a buffer zone to ensure that all air molecules are pumped out so as to achieve a vacuum environment. At Q3, the mass scanning mode is being selected. The voltage is ramped up gradually, which allows the different ions to pass through and hit the detector. This setting allows the scanning of all peptides present in the sample. Now, the DC voltages which are specific for each peptide have been determined. To perform peptide sequencing, the experimenter needs to choose the peptide to be sequenced. This is done by adjusting the settings of Q1 to single mass transmission mode and setting the DC voltage to a specific value that only allows the peptide to pass through and not the rest. In this case, the blue peptide passes through and reaches Q2. At Q2, the peptide is then accelerated to move at high kinetic energy, colliding with inert gases such as nitrogen. This causes the peptide to be broken down into random fragments, which will pass through Q3 and reach the detector. This process is known as collision-induced or activated dissociation, CID or CAD for short. Let us assume that this peptide that was selected for CID or CAD has the following sequence. After random fragmentation by inert gas, the following possible fragments of B and Y ions can be generated. B ions can be considered as N-terminal fragments, whereas Y ions are C-terminal fragments. For simplicity's sake, let us consider only the B ions. In principle, each amino acid has a certain mass, or MZ ratio. The longer the fragment, the heavier its mass will be. The first fragment contains only the amino acid serine. Not only should we expect its mass to be the smallest, it should also correspond to the value of serine. When we consider the next fragment, it consists of two amino acids of serine. Therefore, the value should increase by the mass of another serine molecule. When we consider the third fragment, it now contains lysine. Therefore, the value should increase by the mass of a lysine molecule. In this manner, we can determine the amino acid sequences of any given peptide fragment. Once all peptides have been sequenced, a database search can be performed to see if it matches with the sequences of any proteins in the database. The example shown here has been extremely simplified as we are considering only the B ions. When we incorporate the Y ions, another spectrum can be obtained as shown. In reality, these two spectrums are shown together and can be quite complex if the analysis was done manually. There exist computer softwares and algorithms to perform such search. I will hand my time back to Darren, who who will explain the results that we generated from a set of data provided to us.
Next, we will now look at the results generated from a set of data provided to us. A mascot search was performed to identify the proteins which were carbamylated. Based on that search, we generated an Excel spreadsheet. The following parameters were used. The enzyme entered is trypsin, and the maximum miscleavage is 2. The fixed modification is carbamylomethyl. This is because DDT was added to denature all the disulfide bonds present in the peptide, after which, to prevent the reformation of these bonds, iodoacetic acid was added to perform acylation. This resulted in the formation of carbamylomethyl group to all cysteine residues. Since the addition is applicable to all cysteines, it can be treated as a fixed modification. If it was put as a variable modification, it would increase the computational time, as the software would not assume that all cysteine residues have this modification, and therefore needs to consider the different permutation. Next, the variable modifications we entered is deamination, oxidation, and carbamylation. Deamination and oxidation are selected as these are common modification. Peptide mass tolerance was selected as 10 ppm. This is a measure of the mass spectrometer's ability or resolution to precisely report the peptide masses. If this value was larger, more hits may be generated, but these hits may be false positives. If the value is too low, then perhaps even the actual protein might not be identified, or identified with a low score. The fragment mass tolerance was set at 0.8 Dalton. The mass values were set to monoisotopic, and the instrument used was ESI QUAD TOF. From the mascot search, several columns had been generated. This column gives the protein description. This gives a brief description line of the protein. Each of these proteins have an accession number which can be entered to access more details of the protein. Importantly, to identify the proteins which are present in a sample with a high degree of confidence, we need to look at the PEP score. The PEP score can be understood as something similar to the confidence level. A PEP score more than 70 would imply that the hit is significant, and its corresponding expectation value, which is similar concept to the p-value, is lower than 0.05. In this column, we can see the variable modification which the peptide has been subjected to. The peptide sequence column also shows the amino acids that has been carbamylated. This information is extremely useful because a modification to the amino acid can affect the structure and therefore the function of the protein. Knowing where this modification occurred can better help us understand its effects. From here, we try to identify proteins which are carbamylated. Our selection criterion was based on the PEP score, and proteins with a score above 70 are selected. This spreadsheet has already been sorted by PEP score. From the spreadsheet, we have identified some possible proteins which were carbamylated. This is shown here. This list is certainly not exhaustive. Proteins which are involved in the cytoskeletal network, the basal lamina, and those that regulate apoptosis are some examples of carbamylated proteins. These proteins can be explored further in detail in future experiments to understand the physiological effects or consequences of carbamylation on these proteins. We shall focus on two proteins, clusterin and apolipoproteins. These two proteins are involved in an Alzheimer's disease, or AD for short, and this will be explained by Reuben.
AD is a neurodegenerative disorder that results in progressive cognitive impairment, including dementia, personality changes, and memory loss. It could eventually result in the death of the individual. Patients suffering from Alzheimer's disease will experience extreme shrinkage of the hippocampus, extreme shrinkage of the cerebral cortex, and the entorhinal cortex. A key distinguishing feature of AD patients is the formation of the amyloid plaques and the tau tangles as shown here. From our results generated earlier, two proteins have been identified which could be carbamylated, namely apolipoprotein and clusterin. It has been shown in literature that these two proteins play important regulative roles in amyloid plaque formation and prevention. More research is required to elucidate the mechanism of action of these two proteins in relation to amyloid plaques. Here, we postulate that carbamylation could perhaps play a role in this regulation. This diagram depicts the formation of the amyloid plaques in AD patients. Amyloid fibril formation is a multi-step process which starts with the cleavage of the amyloid fragments from the transmembrane amyloid precursor protein and the misfolding of alpha and beta monomers to form various structures. Amyloid fibrils are unbranched rigid fibers of protein. Proteins that form amyloid are diverse with no homology to each other, although all form an ordered beta sheet interacting structure made up of protofilaments that associate in bundles to form fibrils. From these fibrils, amyloid plaques are formed. A significant number of apolipoproteins such as ApoA and ApoE have been implicated in amyloidogenic diseases or have been shown to form amyloid. Human ApoC2 is mostly associated with very low density lipoproteins, but it plays an important physiological role as an activator of lipoprotein lipase. When associated with polar lipids, ApoC2 adopts an alpha helical conformation. However, under lipid-free conditions, ApoC2 self-associates to form twisted, ribbon-like fibrils showing all characteristics of amyloid. The diagram on the right shows an electron microscopy image of ApoC2. From the diagram, the flat, ribbon-like morphology of the fibrils can be observed. Therefore, to summarize, the aggregation of ApoC2 causes the amyloid plaque formation in the brain, which gives rise to Alzheimer's disease. However, research has shown that amyloid formation by ApoC2 is inhibited by clusterin, which is a heterodimeric protein. From our results obtained earlier in the Excel spreadsheet, we know that clusterin can be carbamylated. However, the question which now arises is, will carbamylation of lysine residues in clusterin alter the structure and therefore function? We can postulate that since clusterin is a heterodimeric protein, perhaps carbamylating one of its monomers may prevent the two protein molecules from dimerizing. From this, we can understand that if the normal function of clusterin is perturbed, it would result in the formation of these fibrils. Therefore, it will be interesting to investigate the physiological effects if clusterin is carbamylated. Alternatively, we also postulate that carbamylation may also occur on the ApoC2, preventing its binding with VLDL. If you recall earlier, we mentioned that ApoC2 binds to VLDL, and this prevents the self-association of ApoC2. Studies have shown that an enzymatic modification known as citracromylation of a critical lysine residue, specifically at the 680th position in apolipoprotein B100, abolishes its ability to bind with apolipoprotein A. What is significant is that citracromylation also occurs at the amino group of the lysine residue, which is the same target group for carbamylation to occur. Hence, we also postulate that carbamylation of the ApoC2 can disrupt its structure and prevent its binding with VLDL. This will allow the ApoC2 to self-associate and thus form the fibrils. Therefore, through this proteomics approach, we have generated a testable hypothesis which would require further investigation. These examples indeed show the importance of understanding carbamylation and how it is involved in disease initiation and progression. In conclusion, carbamylation is an irreversible non-enzymatic PTM and DPM. Carbamylation occurs due to high levels of urea, activity of inflammation, and smoking. It is related to diseases such as cardiovascular disease, renal insufficiency, and arthritis. It may possibly be related to neurodegeneration and formation of amyloid plaques. Further investigation of the effects of carbamylation on ApoC2 and clusterin are required. We have come to the end of our presentation. Thank you for your time.